Let's go over some of the basic concepts of the microscope. Let's start with the total magnification. The total magnification of a slide that we are viewing is determined by multiplying the power of the ocular lens, which is 10x always, by the power of the objective lens you are using. For example, you are viewing a slide using the 4x objective lens. What is the total magnification? You will multiply 4x by the 10x, which is the power of the ocular lens. The answer is 40x. Question: you are viewing a slide using the 10x objective lens. What is the total magnification of the slide? The answer is 100x. Next, we have the field of view, which is the circular area visible through the ocular lenses. The field of view decreases in size as the magnification increases. For example, when you're viewing this slide under the microscope using the 4x objective lens in place, you'll be able to see the entire specimen. Whereas when you view the same slide via the 40x objective lens in place, you'll only be able to see part of the specimen, because we will zoom more on the specimen using the 40x. Question: which objective lens has a greater field of view? Is it the 10x or the 40x? The answer is the 10x. Moving to resolution, which is the ability to distinguish two very closely spaced objects under the microscope as separate points, is known as resolution or resolving power. Moving to the depth of field, which is the depth through which the specimen is clearly in focus. If we examine the colored thread slide, here we have a yellow, blue, and a red thread. At 4x, we will be able to see all three threads in focus at the same time. Whereas if we examine the same slide at 10x or 40x, we will only be able to see one of the threads in focus. Why? Because the depth of field will decrease as the magnification increases. Question: which objective lens has the greater depth of field? Is it the 100x or the 40x? The answer is the 40x. Moving to the working distance, which is the distance between the bottom of the objective lens and the slide on the stage. This is the working distance for the 4x objective lens, whereas this is the working distance for the 10x objective lens. We can see that the working distance will increase as the magnification power decreases. Question: which objective lens has a greater working distance? Is it the 10x or the 40x? The answer is the 10x. The virtual image is the reversed upside down image relative to the specimen. So for example, if we are viewing a real image of the printed letter E, which looks like this, under the microscope at 40x, the virtual image will look like this, upside down, reversed. Moving to parfocal, which is very helpful for a microscope to be parfocal. Why? Because let's say I'm viewing this slide under the microscope via the 4x objective lens. I had my slide in focus and clear. Then when I switched to 10x, which is higher magnification, the slide remained in focus. This is the parafocal, which is when the slide stays in focus at a higher magnification once it is properly focused at a lower magnification.